This section of notes is on another cell division process called meiosis. The purpose of meiosis is to form gametes. The gametes are special cells. They're the egg and the sperm cells. And these cells will have the haploid number of chromosomes. So almost all of our cells divide by mitosis, but there's two special cells, egg in females, sperm in males, that are produced by this special process alone, meiosis. There's some essential vocabulary that we need to know before we get into meiosis. First, we have homologous chromosomes. Homologous chromosomes are a matching chromosome pair. They have similar size, shape, and centromere position, and they have similar but not necessarily identical genetic information. So, for example, a homologous pair of chromosomes may have the genetic information for dimples, but one chromosome might say that you are supposed to have dimples, while the other chromosome might say that you're not supposed to have dimples. So it'll have information for the same traits, but the actual information about the trait could be different. And in our next unit on genetics, we will eventually talk about if there is conflicting information, what do you end up having as your trait? Homologous chromosomes each comes from one parent. So in a pair, one of the chromosomes came from dad and one of the chromosomes came from mom. Homologous chromosomes mean diploid, and they're also called a tetrad. The prefix tetra means four. And the reason homologous chromosomes can be called a tetrad is because we have four chromatids that make up that homologous pair. Haploid is abbreviated N. Haploid is a cell that has half of the chromosomes, meaning only one from each homologous pair. So homologous pair is diploid or 2N. Haploid is half of that, or just N. So haploid versus diploid. Haploid means you only have one set of chromosomes. In humans, our haploid number is 23. And in humans, gametes, our sperm and our ova, are haploid. Diploid means that we have two sets or that homologous pair of chromosomes. In humans, the diploid number is 46. And in humans, that's all of our body cells. Everything other than our gametes are diploid. So an overview of meiosis. Meiosis is cell division for the purposes of reproduction. Meiosis produces the gametes, or the sex cells, which are sperm and ovum. In meiosis, one parent cell goes through two divisions, and that ends up resulting in four daughter cells. And the daughter cells that are produced are completely unique. They are unique compared to one another, and they're unique compared to the parent cell. And in terms of chromosome number, they have only half the number of chromosomes of the parent cell, which makes them haploid, or N for the abbreviation. So in general, the stages of meiosis include interphase. So the parent cell will still go through G1, S, and G2, and then it'll go into the first half of meiosis, which we call meiosis 1. Meiosis 1 will start with that diploid parent cell. It will separate the homologous pairs and result in a haploid cell. So it has half the number of chromosomes, but each of those chromosomes are still made up of two sister chromatids that are still connected. Those two cells will then go into the second half, which is meiosis 2. Meiosis 2 then separates the chromatids and ends with four haploid gamete cells. These cells do not re-enter the cell cycle. That is it for meiosis, and those haploid gamete cells are then ready to be used in fertilization. So now the specific stages of meiosis, starting with meiosis 1. So the very first stage is prophase 1. The chromosomes condense and become visible for the first time, and the nuclear membrane disintegrates and the nucleus disappears. There's two very unique things that happen in prophase 1. One of those is called the homology search. The homology search is when the homologous pairs actually find each other and come together. And so they are physically near each other in the cell. After the homology search, then we have crossing over. In crossing over, the homologous pairs exchange pieces with one another. So the chromosome that came from mom will break off some pieces and exchange those with the homologous partner of the chromosome that came from dad. What this does is it mixes up the DNA even more than meiosis is going to end up doing. So here you can see the homology search on the top when the chromosome pairs find each other, and you can see crossing over on the bottom where the chromosomes actually exchange pieces with one another. The homology search and crossing over increase the variety that we're going to have when we eventually get our cells divided and end up with our gametes. Metaphase 1 is when the homologous pairs line up at the metaphase or at the equatorial plate. So the homologous pairs line up with their partner. What happens here is something called independent assortment. And what that means is that it's completely random how the chromosome pairs will line up during metaphase 1. We cannot predict how it will occur. We don't know the organization or the arrangement that they'll end up being at when they end up lining up with their partner at the equatorial plate. So if the parent cell has 6 chromosomes, so if the diploid number is 2n equals 6, there are many different arrangements of how these 3 homologous pairs can line up during metaphase 1. And you can see the possible arrangements at the bottom. Arrangement 1 versus arrangement 2 versus arrangement 3 versus arrangement 4. It's completely random how those chromosomes will end up pairing up. Now, the more chromosomes that are in the parent cell means that there's more possible random arrangements that could occur in metaphase 1. So if we have 6 chromosomes, 3 pairs that could have 4 different arrangements, just imagine how many different arrangements we could have with our 46 chromosomes or our 23 pairs. So independent assortment is another source of variety, another way that we're going to guarantee the gametes that we end up with are very unique. So then the next stage is anaphase 1. And in anaphase 1, the spindle, which has attached onto those centromeres of the homologous pair, will separate the tetrad. So what gets separated are the homologous pairs. The spindle pulls the separated pairs to opposite sides of the cell. One thing that's important to know at this point is that the centromere that attaches the individual chromatids is still intact. So we've separated the pairs, but not the chromatids. In telophase 1, the chromosomes gather at opposite sides of the cell and nuclei reform. And then in cytokinesis 1, the cell splits. We end up with 2 haploid cells. Each cell has only 23 chromosomes, but each chromosome is still made up of 2 chromatids. So that's why we need a whole nother half of meiosis, because we now need to separate those chromatids. So the second half of meiosis begins with prophase 2. In prophase 2, the chromosomes are visible, the nuclei of each cell disappears, but there's no homology search or crossing over. And that's because we no longer have homologous chromosomes. The homologous chromosomes, those pairs, were separated in anaphase 1 of meiosis. So now we have single chromosomes that are made of 2 chromatids. In metaphase 2, the chromosomes line up single file at the metaphase or equatorial plates. They line up single file because they no longer have a pair or a partner. Anaphase 2 is when the chromatids are pulled apart at the centromeres, and they're pulled to opposite poles of the cells by those spindle fibers. In telophase 2, the chromosomes gather at opposite sides of the cell, and nuclei reform around those chromosomes. And then finally, in cytokinesis 2, those 2 cells split into 4 haploid cells, and these are the gametes, or the sex cells: egg and female, sperm and male. So here are animations that are showing the various stages of meiosis, starting with early prophase 1, metaphase 1. Metaphase 1 there is showing all the different possibilities that could occur due to independent assortment. Anaphase 1, telophase 1, cytokinesis 1, and then the second half, prophase 2, metaphase 2, anaphase 2, and telophase 2 with cytokinesis 2. This slide shows an interactive meiosis activity. This is something that if you did not try yet, you should definitely go back into the meiosis slideshow and go through this interactive activity, because it shows you what happens in the different stages of meiosis. This is just showing how the chromosome number changes during meiosis, how we start with a haploid parent cell. In humans, that means it starts with 46 chromosomes, and how through the 2 divisions of meiosis, we end up with haploid daughter cells in humans having only 23 chromosomes. This is showing that same information as well, how the chromosome number changes throughout the stages of meiosis. And this again is also showing the chromosome number situation in meiosis. This video reviews the stages of meiosis.
Have you ever wondered how 2 siblings can have the same mom and dad and still look so different? Well, today we're going to talk about a process that makes that possible, a process called meiosis, not to be confused with mitosis, which sounds unfortunately similar. Mitosis makes identical body cells, like your skin cells and stomach cells. Recall from our mitosis clip that since it makes identical body cells, mitosis is important for growth and for repair of damage, or to replace worn out cells, but not meiosis. Meiosis is a process that contributes to genetic variety. Meiosis also doesn't make body cells. It makes sperm and egg cells, otherwise known as gametes, the fancier word. You might recall that humans have 46 chromosomes. That's how many chromosomes most body cells in your body have. But there are some human cells that don't have 46 chromosomes. Human sperm cells and egg cells have 23 chromosomes. Why the number difference? Well, if a sperm cell has 23 chromosomes and an egg cell has 23 chromosomes, when they come together, that makes 46 chromosomes. That will allow a newly formed fertilized egg to develop into a human. Meiosis is what we call a reduction division because you have a starting cell that has 46 chromosomes, and your ending cell, the sperm and egg cells, have only 23 chromosomes. Before we can start getting into the stages of meiosis to make gametes, we have to remember what happens before meiosis can even start. Actually, this also happens before mitosis. It's the stage known as interphase. And if you remember interphase, it's when the cell is growing, it's replicating its DNA, it's carrying out cell processes, and just like mitosis, interphase happens before meiosis even going to start. So the starting cell has 46 chromosomes, and you have to duplicate those chromosomes in interphase before meiosis starts. That basically means you're duplicating your DNA since chromosomes are made of DNA and protein. Ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes because the sister chromatids are still attached and we are actually counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture. But you went from 46 to 92 chromatids. Little tricky there. We have a detailed video that explains these chromosome numbers before and after replicating in interphase that can be useful for meiosis. Okay, so interphase checklist done. Now we can move into meiosis. You might remember the mitosis stages PMAT. The P was for prophase, the M was for metaphase, the A for anaphase, and the T for telophase. And the good news is that in meiosis, you still use those terms. But because meiosis is actually a reduction division, you're going from 46 chromosomes to 23, which means you actually divide twice. So instead of mitosis, where you divide one time and do PMAT one time, in meiosis, you're going to divide twice and therefore do PMAT twice. Because of this, in meiosis, you put numbers after the phases to indicate whether you're in the first division or the second division. So let's dive right in. So let's start with the very first step, prophase one. One thing I like to remember about prophase is pro, this actually means before, and it kind of helps you remember that it comes before all the other stages start. This is where the chromosomes are going to condense and thicken. They are actually going to line up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. They're going to match up. It is during this prophase one that this amazing process occurs called crossing over. Now, I know crossing over probably sounds like something very different, but this is actually really awesome process because this is when the chromosomes, they're lined up in homologous pairs and they have this way that they can transfer their genetic information and exchange it between each other. It's kind of like these chromosomes flop over each other and they do a little genetic information exchange here. It makes for what we call recombinant chromosomes, which can eventually contribute to the variety that we were mentioning that siblings can have even when they have the same parents. More about that later. Now we move into metaphase one. In metaphase one, think of the M as standing for middle. The chromosomes are now going to be in the middle of the cell. It's a little bit different though from mitosis because these chromosomes are going to be in pairs in the middle of the cell. So it's not a single file line, they're in pairs in the middle. Now during anaphase one, think of A for away because the chromosomes are going to be pulled away by the spindle fibers. Then we end with telophase one, where you have two newly formed nuclei, and it becomes obvious that you're going to end meiosis one with two new cells. Cytokinesis follows with splitting the cytoplasm. But we're not done yet. On to meiosis two. The very first step in meiosis two is prophase two. It's not going to be nearly as eventful as it was in prophase one, though, because they're not going to have homologous pairs of chromosomes. They also are not going to have this amazing process called crossing over. That doesn't happen again in prophase two. You have your chromosomes, and the spindles are starting to form like they did in prophase one, but prophase two is just not as eventful of having that process of crossing over. In metaphase two, remember, think M for middle, the chromosomes are going to line up in the middle. This time, though, they are in a single file line. They are not in pairs like they were in metaphase one. Anaphase 2, remember A for away, but this time it's the chromatids that are going to be pulled away by the spindle fibers. In telophase 2, you can see the nuclei reforming, and you can also see that the two cells have divided. There's going to be four cells forming. Cytokinesis will follow to completely split the cytoplasm. Now, keep in mind that meiosis in males produces sperm cells, and in females it produces egg cells. Because of independent assortment and also crossing over, you're going to have variety. For example, in a male, the four sperm cells that are produced each time, they are all different from each other. They're also different from the starting cell because the starting cell had 46 chromosomes and the ending cells only had 23. So they are not identical to the original and they are not identical to each other. This is going to lead to variety, a reason why two siblings with the same parents can look different from each other. They still developed from a unique egg and a unique sperm cell that came together. One last thing to think about. Scientists are often looking into the process of meiosis because sometimes the chromosomes don't separate correctly. It's called non-disjunction when a cell can receive too many or too few chromosomes in the separation. This contributes to some genetic disorders, which is something scientists continue to study.
So if we compare meiosis and mitosis, there are some key differences in the processes and specifically in the stages. Here we have a chart that shows that they differ in the divisions, whether they have independent assortment, synapsis, which means do they find their homologous partner, crossing over, the outcome, ploidy, which refers to how many chromosomes do the cells have, use, so what kind of cell does each produce, and the genetic components. Are we producing identical cells or cells with variation? This compares chromosome number between mitosis and meiosis, how they'll both start with a diploid parent cell. Mitosis also ends with diploid daughter cells, while meiosis ends with haploid daughter cells. And here shows a difference between the stages of meiosis, which has the two different halves of it, compared to mitosis. So more specifically, the differences between meiosis and mitosis. A big one is crossing over. Crossing over occurs in prophase 1 of meiosis only. That's the only time we're going to see crossing over. And this is when the homologous chromosomes exchange pieces. Metaphase is very different between mitosis and meiosis as well. In mitosis, the chromosomes line up as 46 individual chromosomes that each have two chromatids. In meiosis, remember we've got two metaphases. In metaphase 1, the chromosomes line up as 23 pairs, and each is made of two chromatids. Whereas in metaphase 2, the chromosomes line up as 23 individual chromosomes made of two chromatids each. The next major difference is in anaphase. In anaphase of mitosis, the chromosomes are pulled apart into two sets of 46 individual chromatids. In meiosis, anaphase 1 will separate the 23 pairs, but each chromosome still consists of two chromatids. And anaphase 2, the chromosomes are pulled apart into two sets of 23 individual chromatids. So this is another chart showing how many chromosomes, how many chromatids we end up with in each of the stages of mitosis, as well as meiosis 1 and meiosis 2. And this video shows us a quick side-by-side comparison of mitosis and meiosis.
In biology, there are often vocabulary terms that sound pretty similar. Chromosome, chromatid, chromatin, transcription, translation, mitosis, meiosis. You probably have encountered this. When I was first learning about mitosis and meiosis, I learned them both separately first. And then I tried to figure out what was the same about them, what was different, why did they both matter. I would try to compare the stages by flipping through images. You know what would have really helped me? A side by side comparison. And that's what this video is. We assume you already have a background of mitosis and meiosis. If not, take a look at our videos on them. But this video is a side by side comparison, presented in a split screen. Mitosis on the left, meiosis on the right. Both of these processes, along with the cytokinesis that follows them to split the cytoplasm, are involved in making new cells. Mitosis results in body cells. Meiosis results in sperm and egg cells, otherwise known as the fancy term gametes. Before we start mitosis and meiosis, let's look at what you start with. Your starting cell in both mitosis and meiosis is diploid, written here as 2N. That means it has two sets of chromosomes. In humans, that's including one set of 23 chromosomes from mom and one set of 23 chromosomes from dad, 46 chromosomes total. During interphase, the cell duplicates the chromosomes. When you duplicate 46 chromosomes, you still say there are 46 chromosomes, as the newly duplicated portion is still attached at the centromere region. But there are actually 92 chromatids. Interphase isn't part of mitosis or meiosis, but it's a really important phase because it duplicates chromosomes before we get started. Just to point out, it's really hard to draw 46 chromosomes. We're going to use 6 chromosomes in our diagrams when we illustrate what's happening because it's much easier to draw and visualize. Oh, and just a fun fact, some insects have 6 chromosomes, like mosquitoes. Unfortunately, I'm not a fan of mosquitoes, but mosquitoes do both mitosis and meiosis. When learning the stages, we give the acronym PMAT, which is helpful for understanding the stages. Both mitosis and meiosis go through these stages, but meiosis goes through them twice and therefore has a number next to each PMAT stage. We're going to show some basic events for each PMAT stage, but please know that there is way more detail to explore than what we can include in this quick video. Prophase and mitosis. Remember that pro can mean before, and this stage comes before the others. The chromosomes are visible. We say they're condensing, which means they are thickening. Prophase 1 in meiosis. Happening here too, but the chromosomes are actually going to match up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. With each pair, one came from mom and one came from dad. In this formation, chromosomes can transfer their genetic information and exchange it between each other. It's called crossing over. It can make for what we call recombinant chromosomes. Metaphase in mitosis. The nuclear envelope which had surrounded the nucleus was already disassembled before metaphase even started. For metaphase, I like to remember the M for middle, because in this stage, the chromosomes line up in the middle of the cell in a single file line. Metaphase 1 in meiosis. The chromosomes are in the middle as well, but they're still going to be in pairs in the middle of the cell, so it's not a single file line. Anaphase in mitosis. I like to think as the A for away, the chromatids are pulled away by the work of the spindles. They are moving to opposite sides of the cell. Anaphase 1 in meiosis. Same thing, but in this case, it's the chromosomes, not chromatids, being pulled away to opposite sides of the cell. Telophase in mitosis and telophase 1 in meiosis. The chromosomes are actually the complete opposite ends, and new nuclei are forming on each side to make two new cells. And they are starting to surround the chromosomes on both sides, as this will eventually form two cells. Cytokinesis follows to split the cytoplasm to complete the actual dividing of the cell. So at the end of mitosis and cytokinesis, we end with two identical diploid cells. In humans, they would both have 46 chromosomes. This is great for organism growth, Growing requires making more cells, after all, or replacing damaged cells. Onto meiosis 2. Prophase 2. Chromosomes are condensing in both cells. It's not going to be as eventful as it was in prophase 1 because they're not going to have homologous pairs and crossing over. Metaphase 2. M for middle, but this time the chromosomes are in a single file line, similar to how metaphase looked in mitosis. Anaphase 2. Think A for away. This time, though, it's actually the chromatids that are getting pulled away. Telophase 2. Chromosomes are at the complete opposite ends, and new nuclei are forming on each side to make these new cells. Cytokinesis will follow meiosis 2 to completely split the cytoplasm. We are now finished with meiosis, and we end with 4 non-identical cells, gametes. Males make sperm cells in meiosis, and females make egg cells in meiosis. These gametes are haploid, meaning they have half the number of chromosomes as the original starting cell. In the case of humans, the resulting cells would each have 23 chromosomes. By the way, when a sperm and egg cell combine, it results into a diploid cell, a fertilized egg, otherwise known as a zygote, which will then start a series of divisions using mitosis to give rise to a brand new organism.
Next, this video goes through a comparison of the chromosome numbers in mitosis and meiosis.
If you have ever studied mitosis or meiosis, chances are the whole chromosome number thing, it's crossed your mind. I mean, how do you start out with a cell that has 46 chromosomes, and then it does mitosis, and then you end with 2 cells that each have 46 chromosomes? Or in meiosis, you end up with 4 cells that each have 23 chromosomes. It's like, here's a chromosome, there's a chromosome everywhere, chromosome, but how? Well, this video is going to explain the chromosome numbers in these processes. And just a reminder, human body cells have 46 chromosomes, and human sex cells, also called gametes, they have 23 chromosomes. Now, chromosomes, as you know, they're made up of DNA and protein. The DNA is highly condensed, tightly wound. That's really important because it means that when the cells divide and they have to move that DNA over, it's easy to do that when the DNA has condensed into chromosomes. It makes DNA so portable and organized, which is great in processes like mitosis and meiosis because when you make new cells, you also have to get the DNA into those cells. This is a chromosome. This is also a chromosome. The difference is that the first one only had 1 chromatid or copy, and the second one has 2 chromatids or copies. The area in the middle of the chromosomes, this is called a centromere, and the centromere is where the spindle is going to attach. That's kind of a big deal because that's how you're going to move the chromosomes during any division process. Now, generally, when you count chromosomes, you can count them by the number of centromeres present. Now, interphase is a step that happens one time before mitosis and meiosis can start. And in interphase, this is where we duplicate the chromosomes. So first we start with 46 of these chromosomes, which is how many chromosomes are found in human body cells. And as you can see in our little picture, they kind of look like little sticks. But after duplicating in interphase, we still have 46 chromosomes technically, but they have 2 chromatids. We call them sister chromatids, and they're held together at 1 centromere. See, as long as those chromatids are still attached at the centromere, we still count 46 chromosomes, but they're duplicated. And if they weren't attached like this, I mean, technically you could say there were 92 chromosomes. But right now in this picture, there are 46 chromosomes and 92 chromatids. Here's a little chart here because I love charts. So is there a time in mitosis when there are 92 chromosomes? Well, technically, you could argue that anaphase, when the sister chromatids are separating and they're no longer attached, you could see in this picture there could be 92 chromosomes because they're not attached during that one phase. And that's why our chart has a little asterisk here. Although we still usually refer to them as chromatids during this process of cell division. So at the end of mitosis, those chromatids separate and you end up with two cells after mitosis that each have 46 chromosomes. Those chromosomes kind of look like little sticks in this picture. The two daughter cells, if you look, are identical to the starting cell in mitosis. Now let's take a look at meiosis. We have 46 chromosomes in the starting cell, and right now they each have one chromatid, just like with mitosis. And they duplicated interphase, and we still have 46 chromosomes now, and there are 92 sister chromatids, just like we started with in mitosis. But at the end of meiosis 1, you have two daughter cells, and they each contain 23 chromosomes with 46 chromatids. At the end of meiosis 2, you have four cells that each have 23 chromosomes, 23 chromatids. Just a reminder that in mitosis, the resulting two cells are identical. So they have the same number of chromosomes as the original cell. So the two daughter cells after mitosis have 46 chromosomes and they're identical. But in meiosis, it's a reduction division. The four daughter cells, they have half the amount of chromosomes as the original cell. That's why each daughter cell after meiosis has 23 chromosomes. So we hope that clears it up a little bit for you. Now, just remember that chromosome counting, it has a lot to do with the way we count chromosomes and this definition of chromatid.
So we've talked about the general process of meiosis, but meiosis does differ in males versus females. We know that meiosis in males makes sperm, meiosis in females produces eggs or ova, but there are also a few other differences. So we'll start with males. Meiosis in males is called spermatogenesis, and this is the production of sperm which takes place in the testes. The production of sperm does not begin until a male is at puberty, and once his body starts producing sperm, that will continue his entire lifetime. So males potentially have the ability to father children from puberty all the way until they die. In meiosis of males, all four cells that are produced become functional sperm. Sperm are the smallest cells in the human body. They consist of cytoplasm, mitochondria, and flagellum. The flagellum is the tail that allows them to swim. The mitochondria gives them the energy they need to be able to swim. And the cytoplasm is full of nutrients that allow their mitochondria to keep producing the energy. Sperm are mobile, meaning they're able to move. They swim with the flagellum. When they're released from the testes, millions are released at a time. And given the right conditions, a sperm cell can survive up to 48 hours. Now, meiosis in females is called oogenesis. So this is ovum production that takes place in the ovaries. Oogenesis actually begins before birth. So a female begins egg production before she's even born. But then it kind of is at a standstill. Once the female reaches puberty, then the maturation of the egg continues. And about one ovum matures every 28 days. Occasionally, there could be more than one ova released, and that would be how we end up getting fraternal twins. So this one ovum every 28 days continues until a female reaches menopause. Menopause is when the body stops maturing eggs, when the hormone levels change, and that occurs somewhere around age 50. There's no set time or set age for that to occur, but it occurs around age 50. Now, meiosis does produce four cells in a female. However, only one of the four cells becomes the functional ovum. One cell receives almost all of the cytoplasm. So it's very unequal division in meiosis 1 and again in meiosis 2. And the reason for that is that ovum needs all of that cytoplasm so it has plenty of nourishment to be able to support a possible fertilized egg. The other 3 cells, since they lack a lot of cytoplasm, those 3 cells are called polar bodies and they end up dying. So even though 4 cells are produced, only 1 is a functional egg cell. The other 3 are polar bodies that die and disintegrate. The egg is stationary. The egg is not able to move on its own. It is able to get from the ovaries to the uterus through the fallopian tubes by cilia in the fallopian tubes. Tiny little hairs actually push the egg along. And the egg can only survive for 24 hours once it's released from the ovary. So oftentimes we hear that a female has a fertile period every month that's about 3 days long. And the reason for that is even though the egg only survives for 24 hours, remember, sperm can survive for up to 48 hours. So if intercourse occurs and there are sperm present 2 days prior to ovulation, when the female releases the egg, those sperm are still alive and can immediately fertilize the egg. And then another 24 hours while the egg stays alive. So those 48 hours that sperm could be present and another 24 hours that the egg will survive gives us that 3-day window of fertility that we sometimes hear about. Here's a side-by-side that shows meiosis in males, which is equal divisions both in meiosis 1 and meiosis 2, resulting in 4 functional sperm, and meiosis in females, which has an unequal division in meiosis 1, another unequal division in meiosis 2, resulting in 1 large ovum and 3 small polar bodies, which will eventually die. So sexual reproduction and development. The whole point of meiosis is to produce the gametes, which then will be able to come together in sexual reproduction and produce a fertilized egg that will then develop into a new individual. So meiosis and mitosis, even though they're very different processes, they depend on each other. We need meiosis to produce the gametes. We need those haploid cells, the sperm and the egg, so that we can have fertilization. Fertilization is when that haploid sperm and haploid egg come together and produces a zygote. A zygote is just a fertilized egg. And if you think about it, N of the sperm plus N of the egg equals 2N. When we have a haploid sperm and a haploid egg come together, we end up with a diploid cell. That zygote or that diploid fertilized egg will then go through mitosis, go through that division, produce somatic cells for growth and development. And then eventually that organism will then be able to do meiosis to produce gametes, to be used in fertilization, to create a fertilized egg that will then go through mitosis. So these 2 processes depend upon each other. And this is just showing a zygote going through mitosis, cells actually dividing. And this is just showing that cyclical connection that we have between mitosis, meiosis, and fertilization. Remember that meiosis reduces the chromosome number from 46 to 23. Fertilization restores the diploid number back to 46, so mitosis can take place.